In our final video focused on protein structure, we are going to talk specifically about enzymes. But before we get there, we'll talk about protein denaturation. So here's the thing about proteins. What you'll hear me say about enzymes is that enzymes, right, are biological catalysts. So if you remember from the general chemistry sections, catalysts can catalyze a reaction without being consumed. But the reason why we put biological in front of this is that enzymes and proteins in general are pretty easily perturbed or pretty easily disturbed in terms of their structure. So for example, you may have heard of people have a fever. Well, when you have a fever, the point of the fever usually is to increase your temperature to help denature proteins, to take them from that folded or the native state to the denatured to the unfolded state. Because if you get a fever, it means that your body usually is battling some sort of virus. And so if you can unfold the proteins for that virus or that living thing, you can essentially kill that living thing. So if you look though, the definition of a fever, like if you're going like the medical grade, anything above, normally we were at 37 degrees centigrade. If you're at 38, technically you qualify for a fever. So 1 degree is enough for our proteins to basically become denatured or not behave the way that they're supposed to. In addition to temperature though, if you change the pH, you can also destroy proteins. Some proteins work really well at low pH versus high pH versus neutral pH. So if you change that pH, they will no longer have the right structure and therefore the right function. So again, when you denature a protein, you are getting rid of some level of protein structure. Now, usually you don't get rid of the primary structure just because that's the covalent linkage. But if you get rid of quaternary structure or tertiary structure especially, you no longer have a functional protein. So it's like when you crack an egg or bake an egg, right? So here we have the native conformation of egg albumin, or we can denature it just by, again, adding heat, cracking it open, doing all those things. So again, you don't get the correct structure, you don't have the right function anymore. So we can put that into talking about enzymes and enzyme function. So enzymes, again, those biological catalysts, which means they can take what we call a substrate, which is the reactant here. So if you take a substrate, you can convert that substrate to a product in this case. And this is done by an enzyme. And the enzyme can do this over and over and over and over again. Here's the thing about enzymes. Enzymes end with ase. This is not to be confused with sugars, which end with ose. So for example, lactase is the enzyme that breaks down lactose. So most enzymes that you'll see in biochemistry, not all of them, but a large majority of them will, based on the name, it'll kind of give you a hint as to what that enzyme does. So lactase, probably related to lactose. So for example, those who are lactose intolerant, they probably lack lactase. They don't have the enzyme that can break that glycosidic linkage between that beta-1,4 linkage between galactose and glucose, for example. So as you can see in the diagram from your book, without drawing it, we're taking our enzyme and we're converting the substrate to the product. So essentially what you can do is you may also see enzyme plus the substrate, and then we have a double arrow, an equilibrium arrow, and we make an enzyme substrate complex. That's what the ES is called. And then we have a single arrow that says E plus P. So what we're doing here is we are binding our enzyme to our substrate. So this, if you notice, nothing has changed here. But this part where we go from enzyme substrate to enzyme plus product, this is the actual catalysis or the chemistry that converts the enzyme that converts the substrate to the product. So we have two steps essentially that we look at when we look at enzymes and their inhibitors. So again, just to define some of these, the substrate is what the enzyme binds to. Those two have to fit together. So you can't, for example, lactase will not interact with glucose. It specifically interacts with lactose. That is its substrate. Okay, and it catalyzes the breakdown of lactose into galactose and glucose. So the product is galactose and glucose, for example. So again, the enzyme must bind the substrate to create this enzyme substrate complex. Then once you bind those together, then you can actually do the chemistry to make the product. Okay, so again, what this is this is enzyme kinetics. So kinetics change the rates meaning that normally a substrate, let's say lactase or lactose can be converted to galactose and glucose, and maybe that takes like 10 years on its own. I don't know exactly what it takes, but for example. But with lactase, now it takes, half of a second. So you decrease the time. So kinetics looks at time, not about the thermodynamics. So we increase the rate of making product from the substrate. So again, the reason why we put the term biological in front of this is because if you change the temperature, the pH, etc., you can actually change how active or inactive these enzymes are. And that's actually, as much as we think that might not be a good thing, it actually is. We don't want all our enzymes on at the same time. We don't want all of them off at the same time. So if they're easily perturbed, we know we can easily turn them on or off depending upon what other forms of regulation we need. So for example, if you don't eat lactose, you probably don't want your lactase just randomly breaking down things. So you can turn it off or you can turn it on. You can also sequester it away. You can change temperatures, things like this. So as you can see from these diagrams from your book, we have an enzyme called pepsin. Pepsin works really well in your stomach to break things down. Your stomach has a very low pH. So pepsin, you wouldn't want pepsin around breaking down proteins in all of your other cells. You only want it working on the food and stuff in your stomach. So pepsin's only active at low pH. So we make pepsin, we ship it to the stomach, then it becomes active in the correct structure. But something like salivary amylase, which we talked about a little bit when we talked about our polysaccharides, that's active in your saliva. So that's around neutral pH because you wouldn't want really acidic saliva and things like this. But when that goes to your stomach, it's not active anymore. So pH can change that activity. And same thing with temperature. A lot of different types of enzymes and proteins and things like this have an optimal temperature, which they work at 37 degrees. You go too high or too low, and it just lacks activity as a result. So again, biological catalyst enzymes, and they're biological because you change things slightly, change pH, temperature, etc., you can change the activity of that enzyme. So we can control that activity again by changing pH, by changing temperature, or we can what we call sequester it. We can just say, hey, protein, pepsin, you're only active in the stomach. I'm not going to ship you to the stomach yet because you haven't eaten anything. So you're just going to stay here in this cell at the wrong pH so you're not working correctly yet. And then, oh, I need it. So then you ship it to the stomach and now it's no longer sequestered. It can do its job. But the other way that we can regulate enzyme activity are inhibitors. So it's important that we go through some information about how to inhibit enzymes, so inhibit to stop it from working. So there are a couple different types of inhibitors that we'll talk about in this class. And so understanding how they interact with the enzyme and how they inhibit is very, very important. So the first type of inhibitor that we have is called a competitive inhibitor. So a competitive inhibitor here, if I add CI for competitive inhibitor, that inhibitor will specifically bind to the enzyme. So it looks a lot like the substrate. But if it binds to that enzyme, you can't make the enzyme substrate complex, and therefore this guy would decrease the conversion of the substrate to the product. So if you make less product, that means your enzyme can no longer do what it's supposed to do. So again, a competitive inhibitor looks very similar to the substrate, which means it can bind the free enzyme, the enzyme by itself. Okay, so it's diagrammed here in the slides that you can see from your book. So here's normally the substrate, but a competitive inhibitor is not substrate, but it looks very similar. So it can sit in what we call the active site. The active site is where the substrate binds and where you do the chemistry to convert to product. If you bind the enzyme in the active site, you can't make the enzyme substrate complex, which means you can't make the product. So therefore, your enzyme is inhibited. A non-competitive is the second type that we will talk about. They bind somewhere else other than the active site. But what they do is they might change the structure of the protein by binding elsewhere. And then what happens is now the substrate doesn't bind as well. And if the substrate can't bind as well, if you're kind of noticing a theme here, if the substrate can't get in there to bind, you don't make product. So in this case, as you can see, here normally we have our normal substrate with our enzyme. Great. But if we have a non-competitive, so non, right, not competitive, so it doesn't bind to the enzyme active site, it doesn't look like substrate, it binds somewhere else. But if you notice, when we bind that non-competitive inhibitor, now all of a sudden the shape of the active site changes and the substrate doesn't fit as well. And if the substrate doesn't fit as well, it can just be let go and you can't turn it over and you can't do the chemistry to product unless it fits just right. So again, you change the shape of the enzyme, you can prevent the substrate from binding or not binding as effectively to effectively shut the enzyme down and prevent it from converting substrate to product. So here's the thing about this. If we want an enzyme to be inhibited, we probably don't want every single enzyme always inhibited in our bodies, but sometimes you might want to say, hey, I have enough of this thing down here. So I know that way back here you were an enzyme to start with. Shut down because I have enough stuff already to deal with. This is what we call negative feedback. Negative feedback says, I have enough of this, shut down the previous stuff to go. So an example is cholesterol. So we talked about cholesterol in the lipids chapter, but there's an enzyme called HMG-CoA reductase. And this enzyme has actually been a target for a bunch of statins. So if you've ever heard of statins like Lipitor, Viator, things like this, what it does is it helps decrease cholesterol levels and things like this if you have too high of those levels. But if you look, cholesterol is what we call a non-competitive inhibitor of HMG-CoA reductase. So if you have too much cholesterol in your system, it can go back and bind elsewhere on HMG-CoA reductase and change the conformation to shut down that enzyme, because the body's saying, hey, it's a way of communicating to say, hey, I have enough of this as we go through. So again, it's a downstream product that we get. And we see this in a lot of different types of metabolic pathways. We see it in glycolysis. If you've heard of glycolysis, the TCA cycle, electron transport chain, your body is constantly communicating to say, hey, ATP, too high, too low, make more, don't make more, etc., depending on what your needs are. Again, a lot of these are signaled. If you think about the higher level signals we receive, like, oh, I'm hungry, oh, I'm not hungry, oh, I need to run away, I need to not run away. All of these help with regulating and communicating. Do I turn enzymes on or do I inhibit them and turn them off? So this is the end of chapter 16. There is a lot of information in these chapters, I know, but please reach out if you have questions or general inquiries about things. Thank you.